Hi and welcome to this demo of ScarfWeb, our cloud-based tool for single-cell genomic analysis. My name is Göran Karlsson. I am the co-founder and head of partnership for Nigen. You can reach me at goran.karlsson@nigen.io. ScarfWeb you find on our homepage nigen.io up here in the top. If you click ScarfWeb, you immediately come to our login page, where you can either sign up for a free account, or if you have one, you can log in and then of course put your login details into our browser and you quickly get into the actual ScarfWeb tool. When you enter ScarfWeb, you land on the project page. The project page is designed for you to easily store and organize your dataset into different projects, as you see here to the left. There's also a folder where you receive datasets from, for example, core facilities, and one where you get data that has been shared with you from another user. However, the project page is also designed for you to collaborate on your datasets with other groups or with colleagues, or even publish the data, but we'll come to that later. So as you see here in the inbox, I have now received 2 datasets. So let's see how we upload those into one of your projects. You click on the inbox. Here we find our 2 files. We can see who uploaded them, and we can also review each file. And when we do that, we can find a description. For example, this is 4 different patients that have been multiplexed and HTO tagged before sequencing. We can add metadata labels, and we select target project. In this case, we take the demo video project. And then down here we have a reminder that we couldn't add any sensitive metadata to the file before the system allows us to accept the dataset. When we do it, the dataset ends up in the project that we selected, and I will do the same for the other file. Go to the video demo, agree with the GDPR, accept the dataset. Now we can find both our recently uploaded datasets in the project we wanted it to be in. If we selected the wrong project, we can easily just move this dataset to another project if you like. Now, one of these datasets are multiplexed. It's this one here. So the first thing we want to do is to demultiplex it. So we just click split dataset and just confirm that we want to demultiplex this sample by their HTO tags. And then we just click confirm down here to the right. And immediately ScarfWeb starts to demultiplex this sample. This takes about a minute. And meanwhile, we can start to analyze our other data set down here, the PBMC sample from blood. So we just push analyze. Now first we're going to set some parameters for analysis. In the first page we can select how many transcripts per cells we will allow for a cell to be included in the analysis, genes per cell, mitochondrial counts, ribosome counts, and so forth. So ScarfWeb sets default values, but we can change them down here if we would like to have exclude some cells that have low transcripts, for example, the same low gene counts, and maybe include less with high gene counts. We can change what we allow for mitochondrial counts, both high and low values, and so forth. The same for ribosomal counts, for example. When we are happy with this, we press continue. So next you can regulate the highly variable genes that will actually make up the heterogeneity of your single-cell data. So you can, for example, decide on the mean expression values for the genes, the corrected variance, and this of course will affect how many highly variable genes you use for your analysis, the minimum number of cells per gene, and so forth. ScarfWeb also suggests a block list for genes that could affect the heterogeneity in a non-relevant way. So for example, by default, ScarfWeb excludes the mitochondrial genes, HLA genes, ribosomal genes, and so forth. But you can decide yourself, you can just remove the blocking of these different genes, so you can add a gene that you want to block yourself down here. So for example, KI67 or something that you think would affect your data in a negative way. When you're done with this, just push continue. And here in the end, you can adjust KNNs, number of PCA dimensions, cluster parameters, and so on. So I won't go into detail on what this means, but it basically affects the tightness of your clusters, the number of clusters, and so forth. When you're ready, you just push Start Analysis, and ScarfWeb will take you back to the project page while your dataset is being analyzed. And this usually takes 1 to 2 minutes, could be a little longer if you have a very big dataset. You know that the analysis is finished when the view icon appears here to the right. But before clicking that, we're going to look at this button here. So this is one analysis. And if you click there, you get the information about the analysis you just performed. So it has all the parameters you decided on, which means that you can repeat this analysis at any time point in the future. You can also redo a different analysis with different parameters and so on as many times as you wish. Now let's go to View here. So if you click View, you get into this analysis interface. So in the middle here you have a UMAP of your data. If you rather prefer T's and E plots, you can get that too. I prefer UMAPs, and I go back to that. So you can click on clusters, and you get the clusters over here. They are not annotated yet, of course. We're going to look into that, how we do that later. So first of all, if we look at this cell metadata up here, we have some quality controls. For example, we can look at genes per cell to try to understand how these clusters have been made, if there are any technical reasons for a cluster, for example. And there are different things you can look at: ribosomal counts, mitochondrial counts, and some other things. Cell cycle phase we can also look at. We can have a G2M score, which you see here. Also note on the right side here you get some plots for whatever you look at. You can look at the cell cycle phase more in a more holistic way. And here we see G2S, M, and G1, and so on. And you can highlight the cells by hovering over these ones over here. If you would like to use this plot for a presentation or publication or something, you just click up here on the download, and it will be downloaded to your computer. Up here you can take a snapshot of the UMAP, and it will end up over here, where you can then download it for any type of use that you're interested in. We will now start to annotate our clusters. So to do that, we first visualize the clusters up here. And if you hover with a mouse pointer above the clusters, you will see the cluster identity here to the right. So there are many ways to annotate these clusters. One is to look for known key genes. So this is peripheral blood mononuclear cells. We can look for myeloid cells, for example, with MPO. And we can see the MPO lights up here in this cluster. You could try some cell surface marker that is known to be expressed on myeloid cells. It highlights the same number of cells. So we can then, with this little data, then suggest that this cluster 4 and 5 are myeloid clusters. So then, of course, we just go in and say, like, cluster 5. We can write it's probably a myeloid. And then we can look for example, B cells with some marker gene. And this quite clearly lights up this cluster 9. And we then label cluster 9 as B cells. And we can go on like this. If you want to know a little bit, get a more broad sense for the gene expression in the different clusters, you can select the cluster, for example, the one we labeled as myeloid, and you get a list of the cluster defining genes for that cluster. There might be some of these genes that you don't know what they are. Then you can go to this left side here. You can click on View at Gene Cards, and then you will be taken with a pop-up window directly to GeneCard's webpage, and you get all the information about that gene that you would need. And if you want to highlight how the different genes here to the right are expressed, you just click on them, and the cells that express these genes will be highlighted in the UMAP. Now, when you have annotated your clusters, you might want to create a figure that motivates why each and every cluster have been labeled as they have. And then we have built in this dot plot function. So here to the left of each gene, you can put them in a dot plot. You can select a number of them. We select, for example, the myeloid ones. Here we go to the B cells. We select the top 4. Like this. And we can take one more cluster. Let's just take cluster 7, and we take the top genes there. And now we're taking 4 of each. And the dot plot, and you can find the icon for it up here to the right, and you click it, and then you come to the dot plot you just created. So here's an example of one of the plots you get in ScarfWeb that you might want to use in some kind of presentation or publication or so on. As you can see here, you can completely customize this dot plot as you want. You can change the colors. I'm going to take the Nigen bluish colors here. And you can also move these genes around. So if you want to change the order of them, you can do so. You can include a maximum of 50 genes in the dot plot, and then it's going to start over. So you can do 50, download it, and then put another 15. You can also select genes individually here. So let's put in CD14, for example. And now we got CD14 here, and I might want to change that to the genes that are for the myeloid. I can just move CD14 up to the other myeloid genes like this, and you can completely customize this. When you're done and you want to download a figure, you can do it here. You can select an SVG or a PNG file, whatever you prefer. Another feature for cluster annotation is to do mapping to a known reference that has already been annotated. And that you can do with the button up here, import labels. You click it. You select the project where you have this reference sample that you want to map to, and I have it in the Lund project. I click the reference sample, so these are peripheral blood cells that I've already annotated. And then I click import labels. And ScarfWeb will then take a few minutes to import the labels from this reference sample. When the label transfer is done, you find the data up here under imported labels. You click it, and then you see that all your clusters have now been annotated based on the label transfer from this reference dataset. Now this UMAP is completely interactive if you use the lasso tool that you find up here to the left. If you click the lasso tool, you can use it to select cells or clusters that you want to know more about and, for example, compare with each other. So let's start by doing a differential gene expression analysis between the myeloid cells up here and the B cells down there. Then we first want to select these clusters. You can do this with the lasso tool here. I, on purpose, select a few purple cells from the other cluster. And if you want to clean that up, you click adjust and you remove all the purple cells. Now you have only the clean green cluster. To save it, you click save, and I will call it myeloid cells. Save it. And then I do the same with the B cell cluster. I select it. I clean it up, remove everything that I don't want, and I save it as B cells. So now I have selected the myeloid cluster and the B cell cluster. But of course with the lasso tool I can also select parts of a cluster. So let's say that I want to compare the myeloid cells with each other. Maybe I think there are some different cell types within the myeloid compartment. I can, for example, select the top of the myeloid cluster like this. I call it then myeloid top. And I might want to compare it with the bottom of the myeloid cluster here. So now I say a few different subpopulations. And there are, of course, other things you can do. You can select cells based on genes. So let's say if we take CD14 here, we see that it's expressed in the myeloid cluster, but not equal, so there's some heterogeneity here. So we might want to compare the CD14 high versus CD14 low cells within this cluster. So we select all these cells that we're interested in, and then you can go to adjust. And here we see a histogram of CD14 expression within the cells that we have selected. And I might want to then compare the low expressing cells. So I just remove the cells that have high expression of CD14 and watch the UMAP, which cells you are actually selecting by doing this. So let's take these. These are CD14 low cells. I confirm the selection, and I go into saved, and I call them CD14 low. And now I, of course, want to do the same thing again, but this time select the CD14 high. So this is, of course, a little bit like a FACS analysis for those who know how to do that. You gate the cells based on the expression of a gene or a cell surface marker or whatever you're interested in. And when it comes to CITE-Seq data, I will show that later, this could be, of course, very powerful to select different subpopulations. I confirm the selection. I go and call this CD14 high. Now I have a few different subpopulations here to the right, and I can now start to compare them, for example, doing differential gene expression analysis. Then we just go here to comparisons. I select the populations I'm interested in. I can compare the myeloid cells with the B cells. Click compare, and the analysis is starting. I can compare the top myeloid with the bottom myeloid, compare, and I can, of course, compare the CD14 low with the CD14 high and compare. And it will take ScarfWeb around about 1 or 2 minutes to do this differential gene expression analysis. When the differential gene expression analysis is done, you can visualize the resulting data either as a table like this. So here we have compared myeloid and B cells. We can look at the list of most differentially expressed genes that are up in the B cells, and you see a lot of typical B cell genes here. You can download these lists as a CSV file, for example, if you want to work more with these differentially expressed genes. You can also visualize the resulting data as a Volcano plot. We do that here for the CD14 low and CD14 high comparison. Again, you can customize this table as you wish before downloading it for use in some kind of presentation. And you can highlight the genes here in the Volcano plot. Not surprisingly, CD14 is the most differentially expressed gene upregulated in the CD14 population. You can continue to highlight the genes here by just clicking the dots and make this plot however you like. Now this is a pretty small data set with only 5,000 cells, and it's only single-cell RNA-Seq that has been done to these cells. So let's go to another more complicated data set to show the final features of ScarfWeb. We go back to the project page, and here in the Demo Lund project, we have a data set with more than 50,000 cells. It's again peripheral blood, mononuclear cells, and also stem and progenitor cells in this data set. I will just show that how we move this, because I want to move this data set to my project that I'm working in at the moment, the demo video. I move it there. It's very simple, and it ends up there straight away. So here we have the data set again. It's already been analyzed, and I'm just going to go straight to the view interface. So here we have these cells, and as you see, it's a much more complicated UMAP. But what is interesting with this data set for us today is that the authors performed CITE-Seq on these cells. So that means that cell surface proteins has also been sequenced. So this is a multi-omics data set. If we want to highlight these cell surface markers with the so-called ADTs, we can either just write ADT in here, and you see all the ADTs that have been used in this experiment, or we can, of course, look for a specific ADT. So CD14 is interesting here again, because it has both been included as a cell surface marker, an ADT sequenced, and also the mRNA for this for the gene of CD14 has, of course, also been sequenced. So we can, for example, compare. Let's do the cell surface protein here. We see the expression here, so we have a lot of CD14 cells in this cluster. and we can just compare that with the mRNA expression, and it's highly overlapping, and in fact we see that in most of the cases where we compare ADTs with mRNA. We have stem cell markers in here, CD90 for example, and we quite clearly see that we have the stem progenitor cells here in this population, at least based on this marker. And we can of course again do the comparisons between CD90 positive cells and CD90 negative cells, also based on this ADT expression, just as we did before in the other data set when we did, for example, for CD14 mRNA. So it would be the same. You select and you adjust, just a lot like a FACS analysis. You can exclude or include the high and low cells, save them and compare the different populations. But that's not what we're going to do now. Instead, we're going to do this, that maybe we're only interested in stem cells. So then we would like, for example, to isolate this population here and do a reclustering of it. So a reanalysis, like it is a new file of only hematopoietic stem and progenitor cells. Then we use the lasso tool again, and we select these cells. And when we're done, we can adjust if we like to remove some cells or some clusters and so on. I'm not going to do that right now. I'm just going to save it as CD90 positive cells, and maybe say perhaps they're stem cells. I save this population. It ended up down here, and now I can extract these cells to a new data set. So if I click this button here, and then I just extract only the cells I selected into a new data set. I'll do that, and the ScarfWeb will automatically take me back to the project page, and we see now that the CD90 positive perhaps stem cells are now being imported into my project. This will take a minute. When the dataset has uploaded on your project, you have to analyze it again. You click the analysis icon, and then you have to go through these 3 steps again. I'm just going to run the default settings for all steps. And the analysis starts. You come back to the project page, and after a minute, your data will have been analyzed. When the analysis is done, you can view your data here, and you come to the UMAP, and now we only look at the cells we selected from the larger data set previously. So again, we have to explore the data a little bit. So let's look at CD90, the ADTs, and we can see here that the highest expression of CD90 is up here. This in itself would indicate that the stem cells might be here, and then differentiate out to the other lineages. Let's look more into that. We take another stem cell gene, AVP. It's also highest expressed here. These seem to be contaminating cells from our rough selection of cells from the larger data set. I will just adjust the UMAP a little bit to see the cells better, like that. So let's see if we can see any signs of commitment into different lineages. We can look for erythroid progenitors using GATA1. And we see that it seems to be erythroid differentiation going in this direction. We can look for a typical myeloid gene like MPO again. And it seems like myeloid progenitors are starting to differentiate down here. So this means, just as we expect from a stem cell population, that we have a kind of continuum, a trajectory towards differentiation into different lineages. So ScarfWeb has a tool to look into these trajectories, and it's a pseudotime analysis that we have here. You start your pseudotime analysis by selecting the trajectory you want to explore. So again, you use the lasso tool. I will select stem cells, and then the trajectory I have discovered in the UMAP towards myeloid cells. I'll select this trajectory here, and when it's selected, I can go to pseudotime analysis up here, or trajectory analysis it's called. We use the current selection. We select the start cluster, which is then cluster 3, and then the end cluster, which is then cluster 11, and I just start analysis. And as usual, it will take ScarfWeb a couple of minutes to create these trajectory analysis. When the analysis is done, it ends up here in the cell metadata. You can click on the trajectory 1, and you find it here in the trajectory analysis, trajectory 1, you open, and what you see here in the visualization is the pseudotime analysis here, and along the pseudotime axis, genes that are up and down regulated are highlighted in this heat map. So for example, this cluster 1 of genes, they are the ones that are high expressed early in the pseudotime and then go down regulated along the pseudotime. And then we have the opposite, of course, in this gene cluster 20. So you can at any time point highlight the genes of each of these steps. So in cluster 1 of genes, we see some typical stem cell markers like EMCN has been published as a cell surface marker for stem cells. We have HLF, a very common marker or gene marker also for stem cells, and NFIB, for example. If we go furthest down in the trajectory and look at cluster 20, we see some very typical myeloid genes like P1 here, for example. So this is a nice way of analyzing a trajectory and getting the genes that are expressed along it. You can at any time point copy this gene list. If you want to use it for some downstream analysis, you can view where this gene signature is mostly active, and then it comes for the entire data set of the UMAP. So these were some of the features that ScarfWeb include, and if you want to learn more about other features or try this tool yourself, I suggest that you create a free account on our website and try the demo data that are there. There are also free uploads for 1 or 2 files that you could use and try your own data. So I will end this demo by going back to the project page and show how this could be used as a portal for the data you have created and analyzed. So we have some functions here next to the different data sets. If you click the right action buttons here, you can choose to do a few things. You can, for example, share the data, and if you click that button, you can share the data to anyone with an email address, or you can share it via link, so someone that gets that link can access your analyzed data. So this is very useful, for example, if you want reviewers to look at your data in an analyzed format. You can send this link in with your submission, and they can access the data in the analysis interface. They can, of course, not do reanalysis. It's kind of a read-only version that they can look at, but they can then explore the data. You can also share it with your collaborators if you want them to see something, or if they want to explore the data that you have generated. Again, they cannot reanalyze the data, so it's just to show the data. Other things you can do, you can transfer the data. Let's say you are a postdoc in a lab and you move to another lab and you want to transfer this data, the ownership of the data, to, for example, the head of lab or to another colleague. You can use the transfer function, and you can also, of course, copy and move and delete. When you're done with your all your analysis and your project and have published it, for example, you can make this data publicly available in then an analyzed format. So this is quite popular among our users. With that, I would like to end this demo. Thank you all for listening, and good luck with your single-cell genomic analysis.